Hello and welcome to today's webinar. Today we have Daniel Sabino with us to talk to us about the eQTL catalog. Over to Daniel.
Hello everyone, good day. Thank you very much for signing on to this webinar about this new resource that we're putting up in collaboration with quite a few people, the eQTL catalog. So in way of background, everybody knows that we're in the era of massive sequencing, these large projects where we're sequencing a large number of individuals, both patients but also controls for a variety of use cases. So there's common disease, rare disease, cancer. Now focusing on common disease, to simplify very crudely, the dominant approach is to do some genome-wide association analysis, or GWAS. The results of these studies are currently being collated by a team of curators here at EBI into the GWAS Catalog. So if you go to www.ebi.ac.uk/GWAS, you get this search window and you can type, let's say, inflammatory bowel disease, and there you'll get a list of SNPs which have been reported to be associated to IBD, along with p-values, odds ratios, etc., etc. Now, these are currently just the top hitting SNPs, so the most significant SNPs as reported by the authors. However, recently GWAS Catalog has been in collaboration with the Open Targets project collecting summary statistics. So instead of getting the top 5, 10 SNPs, you actually get p-values and odds ratios for all the tested SNPs. So this has been quite an intense and exciting work that they've been doing and are releasing little by little. Where you can see that they're either inputting it themselves or getting direct submissions from the authors. Now, we've got all these studies, and for each of these studies, we've got p-values across millions of SNPs. Now you think, well, that's it. All the data is here. We can now just go home. Now, unfortunately, if you want to turn a GWAS study into some therapeutic strategy, there's quite a leap. There's quite a jump that needs to be done, because from association to a variant, we need to go to association to a protein, because for the current drugs act on proteins, not on variants, and therefore we need to find causal genes, not causal variants. Now, if you look into GWAS Catalog, just the top hitting SNPs, these statistics are a few years old, but I really don't think the ratios change that much from one year to the next. You'll see that only a small minority of 7% of top hitting SNPs, which are associated to a phenotype of interest, could be associated, but through the coding sequence, to a putative causal gene. Now, the vast majority of significantly associated variants are intronic and intergenic. Now you'd think, well, okay, we've got a strong association to a variant. It's not on a protein's coding sequence, but it is in the intron of a gene. Therefore, let's just assume it's the closest gene, the nearest gene. Now, one famous example that came out a few years ago is the FTO locus in 2011. A very strong association was found on an intron of the FTO gene to obesity, excuse me. And so the authors naturally reported that the FTO gene seemingly has a strong association to obesity. And a few years later, the Broad Institute, Melissa Klausnitzer and Manolis Kellis in particular, demonstrated that these variants also incidentally appeared to have a very strong association to the expression levels of IRX3, which you can see to the right of this Manhattan plot, and IRX5, which is off the Manhattan plot to the right. So now we've got a much more shaded story. Yes, FTO is the most proximal gene, but these variants appear to regulate the expression levels of IRX3 and IRX5. So it's estimated that in quite a few cases, possibly up to half of them, the nearest gene is not necessarily the one that's being regulated by these intronic and intergenic variants. So what's the solution to this? One approach which has been developed across a large consortia are eQTLs, or expression QTLs. In particular, the GTEx project in the United States is a very extensive collaboration to collect a cohort of roughly 1,000 donors to sample tissues from them and to compute eQTLs off all of them. So this is a tremendous amount of samples, a tremendous amount of experimentation, not to mention all the consenting of the patients and their families and so forth. But now this wealth of data is available. You can go to the GTEx platform, or you can go to Ensemble, and you'll get a view a bit like this one if you go into the gene regulation pages. And so the information that you're getting is that for the gene of interest, BRCA2, so that's it's highlighted towards the middle top of the screen. You can see all the SNPs in its vicinity, and depending on the tissue of interest, so there I displayed 3 tissues out of 44. So there's adipose tissue as well as adrenal gland. You can see all the SNPs roughly within 1 megabase, and you see how strongly they're associated to the expression level of BRCA2. So obviously the SNPs which are directly near BRCA2 are more likely to be correlated to its expression level, but there are some other SNPs a bit farther out which also appear to be distal regulators for the gene. Now, we've got these GWAS studies, we've got these eQTLs. What do we do with it? So one thing that we've been developing in collaboration with the Open Targets project is an analysis platform which would download public GWAS results, perform some standardized search using ontology terms and ontology term matching. We do some LD expansion. This gets us loci of interest. And then we collect all the available data, the eQTLs, but also the high-C, epigenetic data, et cetera, et cetera. And then we try to integrate all this variant to phenotype association into a gene to phenotype association. And so this is something which we just deployed in beta version on Ensemble. That's the Post-GWAS pipeline. Now, this is all great, but GTEx is just one data set. It's probably the biggest human eQTL data set out there, but it's definitely not the only one. We estimate that if you collect all the other data sets, they correspond roughly to the size of GTEx. So there's quite a lot of information out there. So in collaboration with Open Targets, who've been funding this project, we decided to pilot an eQTL catalog. So just like GWAS Catalog collects GWAS results, eQTL Catalog collects public eQTL results. And we're aiming to, by the summer, so by July this year, we'll have processed at least 20 public eQTL datasets, including GTEx. They'll all be reprocessed with a single pipeline, both for expression QTLs and splicing QTLs. So this is really important. We're not just downloading the reports as reported by the authors. Rather, we're running the very same pipeline on all the data sets to remove any analytical artifact. This has been a tremendous and very successful collaboration between the University of Tartu, who are the scientific leads on how to process these data sets correctly, the GWAS Catalog, who are storing all the results, Expression Atlas, who are displaying the RNA-seq quantification, Ensemble, who will be displaying the eQTL scores, and finally Open Targets, who've been helping us with data access applications, as well as displaying our results onto their genetics portal. So this is a pilot, but obviously this is a pilot with the ambitions of becoming a more comprehensive resource. Luckily, luckily data sets are fairly expensive to produce, so we feel that it would be quite reasonable for us to cover all available human eQTL data sets into a single resource, because unlike GWAS publications, which come out every other day, eQTL publications aren't published that often. So how do we process the data? Once again, this has been led very valiantly by our collaborators at Tartu and Caro Alasso in particular. So there you can see a little flow chart where we download the public data sets. We separately process the genotype data, which we QC and impute into VCF files. We also process the phenotype data. So there we're talking about RNA-seq files or microarray files, where we quantify gene expression, transcript usage, exon expression, and transcription events. And there's a bit of statistics, and boom, we store all these statistics into the eQTL catalog and distribute these results via REST API and FTP. So this is a set rather of deployable cloud pipelines. You can go onto our GitHub repo and see the work that we've been doing. We use Nextflow to manage our workflows because we found it to be incredibly useful and reliable. So kudos to the Nextflow team. And so if you go to our GitHub repo, you'll see that there's an RNA-seq quantification pipeline, a normalization and QC pipeline, a genotype QC, and an association testing pipeline. Now, one aspect of using a deployable cloud pipeline is that in the future, some of these eQTL data sets are restricted to us because of the legislation that surrounds them. Some of these data sets come from medical cohorts, and because of how the consent was obtained, they cannot leave the national boundaries of the country. And so our hope is that in the future, if these medical data sets become more common, we will be able to deploy our pipeline locally, respectful of the law, compute our results, and then load these summary statistics into the central database. For a bit of background for all the QTL specialists out there, we've imputed the genotypes using the 1000 Genomes phase 3. We used a bit of genotype filtering. There you can see the parameters filtered out or corrected for a number of covariates, and then we scanned a window of 1 megabase on either side of the gene center. So how do we QC these data sets? As you can see on the left, we produced a number of plots which you can download on the FTP site. You've got gene expression PCA plots and multidimensional scaling, or MDS plots. We also do sex-specific gene expression plots and genotype PCAs. So there on the right, you can see a really nice MDS plot of different tissue types that cluster really nicely and very neatly. So there are some anomalies which can be detected that way. So for example, if you plot out the expression of Y chromosome genes on the Y-axis appropriately, and exist gene on the X-axis, for primary immune cell, you can see that this is a very easy and logical way of distinguishing male from female samples. Now, if you do this on iPS-derived cell lines, you'll see that the exist expression appears to be lost in some iPSC-derived cell lines. So this is already an interesting kind of observation, and that allows us to QC the data sets. Also, we looked at the PCA of genotypes as compared to the 1000 Genomes references. And so there you can see a very clean separation of population, and in purple you can see ALASO 2018 clustering very tightly with mostly European samples, and one of their samples clusters with South Asians. However, on a more diverse study, for example Schmiedel 2018, you can see roughly the same PCA plot, but in this case, these purple dots, which come from the study, are spread out much more broadly, both across different superpopulations, but also between the superpopulations. How do we distribute the data? So the first port of call would be the eQTL Catalog website, which is quite easy to remember, www.ebi.ac.uk/eqtl. So I'll jump off to this website very quickly. So here we go, www.ebi.ac.uk/eqtl, so easy to remember. You get our GDPR banner, which you can dismiss. You get a bit of background information, which corresponds roughly to how I started this presentation. You can see a link to our RTQ. You can send an email, and it'll be received by all the team, and it'll be ticketed and tracked in case it's a long-standing issue. You can look at our methods methods. So this is a longer version of what I told you earlier about our various pipelines and the various methods and so forth. The data access is here. There's various types of data access, a way to access the data. You can either download via FTP either the HDF5 files, so more about HDF5 in a second, but basically these are index binary files which are very powerful to store and access these very large data matrices. But we also have more traditional tabixed, so tab delimited files. They're compressed and indexed. And so these are nifty because of two things. One, you can either just download straight off our FTP a sliver of the data using the tabix utility, or you can just download these entire files, unzip them, and then you'll have a very easy tab delimited file, which is very easy to parse really with any software. And finally is the API access. So this is quite exciting because it's powered by the HDF5 file format I mentioned earlier. So HDF5 is quite useful because it allows you to really extract just a slice of data from the file. So instead of downloading megabases, you just download exactly what you need. So if you go to the URL www.ebi.ac.uk/eqtl/API, you're welcomed by this little response. So obviously this is useful if you're debugging, but programmatically this means that your scripts can quickly peruse through these links and find the useful information. And so, for example, you might want to look at what are the studies that are available. Boom, you click here. And there you get a massive list, a large list of different publications. And so there you'll get, for any of them, you can, for example, ask for Fairfax 2012, what tissues are available. Well, then you click on the link and then you get more information with an ontology link, et cetera, et cetera. So returning to the slides, sorry, I should go back quickly to the main website. Got distracted with the API. We have a list of data sets. This is just for your kind of background information. We also have a very simple license. All the code is available under Apache 2.0, so it's really reusable for pretty much everything. And the data is under CC Creative Commons Attribution 4.0, so incredibly permissive, and you can do quite a lot. And finally, there's the team and our funders, Open Targets, but more about this in a sec. So I've mentioned the website, I've mentioned the API. So as you could see, the API was incredibly reactive. I was just there clicking at random. I genuinely did not script that demo. So one thing that we've done there at the bottom row, you can see a link to an R script. And so this R script allows you to co-localization plots between GWAS studies stored in the GWAS catalog and eQTL studies stored in the eQTL catalog. And so this is something, more about this later, but it's part of our preprint. Another way to access our data is on the Open Targets genetics platform. So here, for example, we've got the profile page for a variant of interest. So in this case, it's RS6740847. So if you scroll a bit down, you can see information about what genes are assigned to this variant. And if you click on the eQTL tab, scroll down, and there you'll see some familiar names such as Fairfax 2012, which we just examined via the API. And so you can see how strongly this variant is correlated or associated, excuse me, to the expression level of the nearby genes here. So that's one way of accessing our data in a nice little interactive browser. Another way of accessing the data is a bit more indirect, but much more informative, is if you're looking at a study of interest. So here we're on this information page about this GWAS study. So this is a publication on Crohn's disease. So looking at this particular variant under that GWAS study, if you scroll down, you'll see some co-localization results. And quite interestingly here, you can see, if you scroll down to QTLs, you can see once again Fairfax showing up and other eQTL data sets from the eQTL catalog that have been co-located with that specific GWAS study. Coming soon, so there I've talked about the Open Targets genetics platform. But in the near future, we'll be matching the work done in eQTL catalog onto the Expression Atlas, which means that for every eQTL catalog dataset, you will find the matching expression results on Expression Atlas. And we will be also displaying, hopefully in the next release, I believe, the same kind of Manhattan plots, which I showed earlier for GTEx. We'll have the same for all eQTL catalog data sets. And obviously we've processed, I believe, 16 data sets, but we will naturally be processing more data sets into the near future. In particular, we'd like to store GTEx. So for GTEx being such a reference annotation, we'll store it under 2 versions. We'll store it with the calculations performed by the GTEx consortium, so matching those published on the GTEx portal. And in parallel, we will provide our recomputed values so that it's homogeneous with the rest of the eQTL catalog. So depending on what you're comparing to, you might prefer to access one version of the dataset or the other, but both should be available. More information. As you can see, we're starting to have a little bit of a social media presence. You can follow our friendly eQTL catalog Twitter account with updates on the various outputs of the group. There's our GitHub repo, so where you can see all our code quite freely accessible. We have a preprint that just came out, which describes all the methods and also describes this script, which performs automatic co-localization using the REST APIs of both GWAS and eQTL catalog. And naturally, there's the website, which I quickly demo to you. So with this, what are the take-home messages? Expression and splicing QTLs are a promising tool to go from GWAS results, summary statistics, to drug targets. We have just released a first batch of data via FTP, REST, and the Open Targets Genetics platform, which you can already access directly with the links that are provided in the slide deck. We'll soon be linked up to the Gene Expression Atlas and Ensemble. And finally, we're aiming to develop a sustainable resource that could keep up with the overall production of public human eQTL datasets. So with this, I'll acknowledge quite many people across. So there's the EBI, European Bioinformatics Institute. There's the Sanger Institute. There's the University of Tartu. And so lots of collaborators from everywhere really converging and helping us produce this nifty little resource. So with this, I'll thank you very much for your attention and take any question you may have. Thank you very much. Okay, so the question, is there a long-term plan to add other QTLs such as protein or methylation? So thank you very much for this question. Yes, in the long term is definitely compatible with what we're trying to do, and I know that there's lots of demand, especially from Open Targets platform partners who are already partners into this project. We don't have any immediate plans, though, to integrate these data sets as the quantification, as we really need to study how to quantify and normalize these data types and make sure that it meshes well with the existing expression and splicing QTL. So to summarize, yes, we've got long-term plans, but not short-term plans.
I'll leave it there for today.
Okay, thank you very much.
Thank you, Daniel. Thank you all for listening.